Okay, goodbye. Okay, now coming to our chapter is a part of microbiology in association with bacteria. Viruses and విషయం కోసం introduction to viruses. Okay, ఈ viruses అనేది నీటి ఉండదు మామూలుగా నీటి ఇది ఐపీ ఉంటుంది వెబ్సైట్ ఐపీ ఉంటుంది వెబ్సైట్ మళ్ళీ ఓకేనా. You know, there are different kinds of microorganisms, including bacteria, viruses, some algae, fungi, and protozoals, etc. So, viruses are also a type of microorganisms, and these viruses are generally the unique group of biological entities. Unique group of biological entities. Entities means acellular smallest organisms. They are the unique group of biological entities. These viruses can infect all types of organisms, means all types of cells. They can infect all types of cells. Animal cells, plant cells, fungal cells, algal cells, and protozoans, even human being cells. అన్నిటి ఇన్ఫెక్ట్ చేస్తాయి, ప్రతి దాన్ని ఇన్ఫెక్ట్ చేస్తాయి. Particularly the viruses which infect the animals are called zoophages. Zoophages. That infect the fungi, they are called mycophages. Myco means fungi. That infect plants are called phytophages. Okay, that infect yeast cells are called zymophages. Like that, all these viruses, they can infect all types of cells. Okay, similarly these viruses are acellular, very important. Viruses are acellular organisms. Acellular, no cellular organization, no cell. Acellular means no cellular organization. So that cell theory is not applicable to viruses, because cell theory explains the cell structure and functions. But here cellular organization is completely absent, so that cell theory proposed by Schleiden and Schwann is not applicable to viruses, because viruses are acellular. Viruses are acellular. Generally, these acellular viruses, they contain nucleic acid and protein. Nucleic acid and protein. Is represented no cellular organization. Is represented by a core of nucleic acid. Surrounding this nucleic acid is called protein. Surrounding this nucleic acid, this is called protein. Okay. The subunits of this protein are called capsomers. Capsomers, because protein part of the virus is also called capsid. Capsid. So every virus is represented by nucleic acid and protein. Nucleic acid core. It may be DNA or RNA. There is no the virus with both nucleic acids, either DNA or RNA. Options are very important. The nucleic acids of viruses are anichasa person. Only DNA, only RNA, DNA and RNA, DNA or RNA. Which one is right? DNA or RNA. Only one type of nucleic acid. There is no the virus with both nucleic acids. Now this protein part of the virus is called capsid. Now this capsid is also contain number of subunits, associated subunits. It is not a single structure. It is formed by the association of many small subunits. They are called capsules. Okay. Similarly, these type of viruses, okay, very important. These viruses, they have no metabolism, okay, no metabolism and no motility, so that these viruses are considered as non-living things, non-living characters, very important, non-living characters of viruses. Okay, what are non-living characters of viruses? They are no metabolism, okay, no motility. They are non-living characteristics of viruses. Similarly, these viruses, they contain continuity, okay, continuity of genetic material, and mutations. Some sudden changes also takes place in this nucleic acid, either DNA or RNA. Continuity of genetic material from one generation to the next, and mutations and replication. Replication means multiplication of viruses, reproduction process of viruses. Replication, multiplication of viruses. So, continuity of genetic material, mutations and replications, they are found in viruses. So that these characters are considered as living characters. So, viruses, they exhibit some non-living characters and living characters. Okay, they exhibit some non-living characters and as well as living characters. Because of this reason, viruses are also called connecting links. Okay, connecting links between living and non-living things. Connecting links between living and non-living things. జీవులకి నిర్జీవులకి ఇవి మధ్య కనెక్టింగ్ లింక్స్ అక్కడ నాన్ లివింగ్ క్యారెక్టర్స్ ఉన్నాయి, లివింగ్ క్యారెక్టర్స్ ఉన్నాయి. ఓకే, సో దే ఆర్ కనెక్టింగ్ లింక్స్ బిట్వీన్ లివింగ్ థింగ్స్ అండ్ నాన్ లివింగ్ థింగ్స్. సో దట్ దీస్ వైరసెస్ దే ఆర్ ఆల్సో కన్సిడర్డ్ యాజ్ infectious particles. Okay, infectious particles are also called infectious virions. And what are those? Virions. Infectious particles are infectious virions. Okay, very important. They are the non-living characteristics of viruses and living characteristics of viruses. So the combination of both living and non-living characteristics. So connecting links between living and non-living things. So that they are also called infectious particles or virions. At the same time, these viruses are acellular, and I already cheppan. They are acellular, no cellular organization. Especially these viruses are, they are intracellular obligate parasites. Intracellular obligate parasites, very important. Intracellular, intercellular, intracellular. These are the organisms which are present inside the cell and cause different kinds of diseases in host organisms. And obligate intracellular parasites. So obligate means host is necessary. These organisms, in the presence of hosts, they act as living organisms. Okay, living organisms. And also multiplication, that is replication, takes place and also cause diseases inside the host organism. But in the absence of host organism, they are present as non-living particles. Non-living particles are virions. Non-living particles are virions. ఇవి ఏంటంటే ఏదైనా ఒక పోస్ట్ దొరికితే, ఇప్పుడు మనం చెప్తున్నాం అనిమల్స్ని అటాక్ చేసే వైరస్ని జూ కాజెస్ అంటాం. ఈ జూ పాజెస్కి ఏదైనా ఒక అనిమల్ కనుక దొరికితే, పోస్ట్ దొరికితే, they behave like living organisms and also attack the host and behave like living organisms and also show replication and also cause some diseases inside the host. But in the absence of relevant host, they are present as non-living particles. They are in the form of non-living particles outside the body of host, but inside the body of host, they behave like living organisms and also replicates inside the host, and also they also cause some diseases. Okay, nowadays the study of viruses, it is established into a new science, a new branch in microbiology, that is called virology. This is called virology. This is the study of viruses. Study of viruses. Study of bacteria is called bacteriology, and study of viruses is called virology. So bacteriology and virology, they include in microbiology. They are the sub branches of microbiology. So nowadays study of viruses is considered as virology. Emantunnandini virology. Okay, this is introduction. Now coming to another point. This is very important. History. ఎక్కడ వైరస్ని ఎలా కనుక్కోవడం జరిగింది అనేటువంటి విషయం కోసం హిస్టరీ. ఓకే, viruses, they cause different kinds of diseases in humans, plants and also animals. Already know some dangerous viral diseases like polio, influenza, AIDS, rabies, ఇవన్నీ కూడా వైరల్ డిసీజెస్. yellow fever, ఇవన్నీ కూడా వైరల్ డిసీజెస్. They cause different types of diseases in human beings and plants and also animals. But these type of viruses, they are not known to the people even after the proposition of after the publication of or after the proposition of the germ theory of diseases. మన తెలుసు ఒక సైంటిస్టు డిసీజెస్ గురించి ఎక్స్ప్లెయిన్ చేస్తూ germ theory of diseases అనే బుక్ రాశాడు. ఎవరు రాశారు? germ theory of diseases, published by scientist Robert Koch. Okay, Robert Koch. Who is author of germ theory of diseases? Robert Koch. Explained the characters and the nature of pathogens in his book Germ Theory of Disease. So actually viruses are, they cause different kinds of diseases from ancient times to even today, but they are not known to the human society even after the publication of Germ Theory of Diseases by Robert Koch. రాబర్ట్ కోచ్ రకరకాల డిసీజెస్ గురించి, రకరకాల ప్యాథోజన్స్ గురించి ఒక బుక్ రాసినప్పటికీ కూడా ఇవి ఉన్నాయనేది ఎవరికి తెలియదు ఆ విషయం. దే ఆర్ నాట్ నోన్ టు ద సొసైటీ ఈవెన్ ఆఫ్టర్ ద పబ్లికేషన్ ఆఫ్ germ theory of diseases by Robert Koch. But in 1892, okay, one of the scientists, Ivanovsky, okay, scientist Ivanovsky conducted extensive research work on tobacco mosaic disease. Tobacco mosaic disease. Okay, conducted research work, extensive research work on tobacco mosaic disease. Already know mosaic disease is a type of plant disease caused by viruses. Mosaic means the loss of chlorophyll in the surface of leaf lamina in the patches, loss of chlorophyll and conversion of green lamina into white color in the patches of the green lamina, that is called mosaic disease. He conducted extensive research work on tobacco plants, tobacco mosaic disease, and this scientist also reported one thing, that is causal agents. Okay, causal agents of this tobacco mosaic disease, they are smaller than bacteriaceae. Smaller than bacteriaceae. Okay? ఈ విషయం అనుకున్నాం. Smaller than bacteriaceae. And also he reported one thing, that is, these causal agents of this disease, they are filterable agents. Okay? Filterable agents. Very, very important. ఈ విషయం అనుకున్నాను. నేను ఏం చేస్తానంటే మొత్తం వ్యాధి కలిగినటువంటి డిసీజ్ లీవ్స్ అన్ని తీసుకొని గ్రైండ్ చేసి దాన్ని ఏం చేస్తారంటే బ్యాక్టీరియాల్ని వడగట్టగలిగినటువంటి ఒక క్లాత్లో దీన్ని వడగట్టే ఏమైంది, ఇది వెళ్ళిపోయింది, దాంట్లో నుంచి ఫిల్టర్ అయిపోయింది. బ్యాక్టీరియాలు ఫిల్టర్ కానటువంటి క్లాత్లో ఇవి ఫిల్టర్ అయిపోయింది. అంటే దాన్ని బట్టి ఏమవుతుంది, దీస్ కాజల్ ఏజెంట్స్ ఆఫ్ డిసీజ్, దే ఆర్ స్మాలర్ దెన్ బ్యాక్టీరియా అండ్ దే ఆర్ ఫిల్టరబుల్ ఏజెంట్స్. ఓకే. Subsequently, one of the scientists, namely Bayes-Rink, okay, Bayes-Rink, scientist Bayes-Rink collected some infected tobacco leaves and also grinded and also filtered and collected the extraction of the leaf. Collected extraction of the leaf. Okay. Extraction of the leaf. ఆయన ఏం చేస్తాడంటే ఫిల్టర్ చేస్తాడు కదా, అట్లాగే ఏం కూడా ఏం చేస్తాడు, ఎక్స్ట్రాక్షన్ తీసుకున్నాడు. పసులు వ్యాధి సోకిన లీఫ్ని గ్రైండ్ చేసి పసులు తీసుకొని నౌ దిస్ ఎక్స్ట్రాక్షన్ ఇట్ ఈస్ ఆల్సో అప్లై టు హెల్తీ ప్లాంట్స్. మరి నార్మల్ హెల్తీ మొక్కల పైన చల్లాడు. అండ్ హీ ఆల్సో అబ్జర్వ్ ఇమీడియట్ ఇన్ఫెక్షన్ బై దిస్ టైప్ ఆఫ్ ఏజెంట్స్. This extraction collected from the infected leaves of tobacco, when it is applied to the healthy plants, immediately these plants are also infected by the same disease. Now, this extraction was named as Contagium vivum fluidum. Contagium vivum fluidum. It was introduced by, this term was introduced by scientist Bayes-Rink. Contagium vivum fluidum. The meaning of this Contagium vivum fluidum means infectious. This is a contagious living fluid or infectious living fluid. Contagium ante contamination, contagium ante infection. Contagious, very, very important. Contagious living fluid. Okay, living fluid, very, very important. దాని అర్థం అది వ్యాధి కల్పించే ఫ్లూయిడ్ అని. కంటాజియస్ లివింగ్ ఫ్లూయిడ్, డిసీజ్ కాజింగ్ లివింగ్ ఫ్లూయిడ్ అని అర్థం. కంటాజియం వైవం ఫ్లూయిడ్. దిస్ వాస్ ఇంట్రడ్యూస్డ్ బై, దిస్ టర్మ్ వాస్ ఇంట్రడ్యూస్డ్ బై సైంటిస్ట్ Bayes-Rink. Okay. Subsequently, in 1935, okay, 1935, one scientist conducted extensive research work on Contagium vivum fluid discovered by Bayes-Rink. దీనిపై పరిశోధన చేసి సైంటిస్ట్ స్టాన్లీ, ఓకే, సైంటిస్ట్ స్టాన్లీ in 1935 conducted extensive research work and also purified the Contagium vivum fluid discovered by Bayes-Rink and also disease causing agents were crystallized. Disease causing agents are crystallized. Okay, disease causing agents are crystallized in the form of small crystals and also named as tobacco mosaic virus. Okay, tobacco mosaic virus. Simply this is abbreviated as TMV. So the actual credit of the discovery of viruses at the first time goes to Ivanovsky in 1892. But the actual credit of the crystallization of these viruses goes to scientist Stanley. In 1935, purified the contagion biome fluid and also isolated some disease-causing crystals from this fluid and also named as tobacco mosaic virus and also awarded Nobel Prize for this pioneering work, scientist Stanley. Okay, దీనికి వైరస్ మొట్టమొదట కనుక్కున్నారంటే వాస్తవానికి ఏవో ఉన్నాయి, బ్యాక్టీరియా కంటే చెడు ఏవో ఉన్నాయని అనుకున్నారు. అవి వాటిని ఫిల్టర్ చేసి చూపించినటువంటి సైంటిస్టు స్టాన్లీ awarded Nobel Prize for this discovery and isolation and crystallization of these viruses. Subsequently, number of scientists are also interested to study the structure of viruses. ఈ వైరసెస్లో ఏముంది అని తెలుసుకునే ప్రయత్నం చేస్తారు. ఒకసారి వైరస్ని బయట తీసాక దాంట్లో ఏముంది, దాని కెమికల్ స్ట్రక్చర్ ఏంటి అనేటువంటి విషయం మీద ఇంట్రెస్ట్ చూపించారు చాలా. సైంటిస్ట్ ఫ్రాంక్లిన్ అండ్ కాన్రాడ్. ఓకే, Franklin and Conrad. Scientist Franklin and Conrad conducted structural, explained the structure of TMB and also analyzed the structure and chemical nature of these TMB. And these two scientists they discovered that is genetic material of this virus, that is as ribonucleic acid. So the discovery of nucleic acid, ribonucleic acid, goes to the scientists Conrad and Franklin. Analyzed this viral particles and also discovered genetic material, RNA, genetic material that is as RNA. So RNA was discovered by Conrad and Franklin. Similarly, different kinds of viruses have been isolated from different kinds of organisms today and also explain their structure and their nature, their chemical structure and physical structure of these viruses by using some advanced technologies like gel electrophoresis and ultracentrifugation. Okay, ultracentrifugation. This is one of the advanced technology, cytological technique, and also X-ray crystallography and electron microscopy. ఇవన్నీ కూడా ఓకే, ఇవన్నీ కూడా అడ్వాన్స్డ్ టెక్నాలజీస్. వీటన్నిటిని ఉపయోగించుకొని రకరకాల వైరసెస్ ఈరోజు పెట్టారు. Different kinds of viral particles have been isolated from different organisms and also analyzed their structure and chemical nature. Chemical studies are also completed, and these chemical studies are completed by using advanced techniques like ultracentrifugation and X-ray crystallography and electron microscopy, etc. ఈ రోజు ఒక సపరేట్ సైన్స్గా డెవలప్ అయింది. తో పాటు ఇట్లాంటి టెక్నిక్స్ ఉపయోగించుకొని చాలా రకాలను మనం ఐడెంటిఫై చేసాం. అటు నుంచి వచ్చే డిసీజెస్ ఐడెంటిఫై చేసాం, వాటి కంట్రోల్ మెజర్స్ కూడా ఐడెంటిఫై చేసాం బై యూసింగ్ ద నాలెడ్జ్ ఆఫ్ సపరేట్ బ్రాంచ్ ఇన్ మైక్రోబయాలజీ, దట్ ఈస్ మైక్రో ఆర్ వైరాలజీ. ఓకే, నౌ కమింగ్ టు అదర్ పాయింట్స్, దిస్ ఈస్ వైరల్ క్లాసిఫికేషన్. ఓకే, వైరసెస్ని ఎలా క్లాసిఫికేషన్ చేస్తాము అనేటువంటి విషయానికి వస్తుంది. Viral classification. You know, the viral classification, it is mainly based on the rules and regulations of ICTV. Viral classification is under the controlling of ICTV. ICTV means International Committee on Taxonomy of Viruses. Very, very important. Okay, International Committee on Viral Classification. International Committee on Viral Classification. This is simply abbreviated as ICT. So classification of viruses is under the controlling of, under the guidelines of International Committee on Viral Classification. Okay, here అంటే ఇక్కడ చూడండి ఇంటర్నేషనల్ కమిటీ ఆన్ టాక్సానమీ ఆఫ్ వైరసెస్ అకార్డింగ్ టు ది స్కీమ్ ఆఫ్ క్లాసిఫికేషన్ according to the scheme of ICTV classification of viruses. The total classification system is represented by only three hierarchical groups, three hierarchical levels. One is family. One is family. Now this family is also divided into some subfamilies. Family. And second one is genus. Okay. And third one is species. There are only three hierarchical levels in the classification system of scheme of ICT. One is family, and other one is genus, and other one is species. Always the name of family. It is ends with the suffix. This is suffix viridae. Viridae. This is the suffix of family. ప్రతి ఫ్యామిలీ పేరు ఇక్కడ మనం చూస్తున్నాం. మాల్వేసియే, సోలనేసియే. ACA is the suffix of the names of families of flowering plants. Like that, here every name of the family it ends with the suffix viridae. Similarly, genus it is suffixed with the virus. It ends with the suffix virus. And similarly, species. It is the species. The names of the species. The names of species. It is common English version. That explains the nature of virus. Virus యొక్క నేచర్ తెలియజేస్తుంది. అది ఏ రకమైన వ్యాధిని కలిగిస్తుందని కానీ దాని యొక్క నేచర్ తెలియజేస్తుంది. సో నేమ్స్ ఆఫ్ స్పీషీస్ ఆఫ్ వైరసెస్ దే ఆర్ కామన్ ఇంగ్లీష్ వర్షన్స్ దట్ ఎక్స్ప్లెయిన్స్ ద నేచర్ ఆఫ్ వైరస్. ఓకే, నేచర్ ఆఫ్ వైరస్. ఇలా మూడు మాత్రమే ఇక్కడ ఉంటాయి. నేచర్ ఆఫ్ వైరస్ మూడు లెవెల్స్ మాత్రమే ఉంటాయి. వన్ ఫ్యామిలీ, suffix viridae, genus virus, and species. This is a common English version that explains the nature of virus. For example, the classification system of AIDS virus, AIDS causing virus. Okay, the systematic position of AIDS causing virus in the classification system, that is according to ICTV, family is Retroviridae. Family Retroviridae, very very important. Retroviridae. మనం చెప్పాం viridae is the suffix. Family పేరుకి viridae is the suffix. Similarly, this genus of AIDS virus, this is lentivirus. Lentivirus. Okay. Retroviridae, lentivirus, suffix virus. And species, this is human immune deficiency virus, HIV. Okay, HIV, human immune deficiency virus. This is the classification system of AIDS causing disease. One family Retroviridae, and two genus lentivirus, and species human immune deficiency virus. That is the systematic position of AIDS virus according to ICTV classification. ఇది మామూలుగా దీంట్లో ఉన్నటువంటి క్లాసిఫికేషన్. కాకపోతే ఇక్కడ ఒక విషయం గుర్తుపెట్టుకోవాలి ఏంటంటే సమ్ వైరసెస్ దే ఆర్ నేమ్డ్ ఆఫ్టర్ ద డిసీజ్ కాజ్. సమ్ వైరసెస్ ఆర్ నేమ్డ్ ఆఫ్టర్ ద డిసీజ్ దే కాజ్. ఫర్ ఎగ్జాంపుల్ polio virus. Polio virus. Okay, virus పేరే polio virus is causal agent of polio. పోలియోని ఏర్పరిచే వైరస్ని పోలియో వైరస్ అని అంటాం. ఇంకా దీనికి ఇక్కడ జీనస్ స్పీషీస్ ఏం లేదు. దే ఆర్ నేమ్డ్ ఆఫ్టర్ ద డిసీజ్ దే కాజ్. సమ్ ఆఫ్ ద వైరసెస్ ఆర్ నేమ్డ్ ఆఫ్టర్ ద డిసీజ్ దే కాజ్. వన్ ఆఫ్ ద ఎగ్జాంపుల్ ఇస్ పోలియో వైరస్. ఏమిది? పోలియో వైరస్. ఓకే, నౌ కమింగ్ టు అనదర్ పాయింట్. This is next point, size of viruses. Okay, size of viruses very very important. Okay na, ఏంటి యొక్క సైజు మనం చెప్పాం, they are smaller than bacterial cells, filterable agents. But what about the particular size of this viruses? The size of viruses it may ranging from highest top most level, that is 300 nanometers long. 300 nanometers long. It may ranging from as long as 300 nanometers, as short as small as 20 nanometers size. This is the topmost spectrum and this is the least spectrum. For example, the topmost, this is the longest virus and this is shortest virus. Longest virus, for example, TMV. TM is the largest, longest or largest. And very smallest and 20 nanometers size, that is RO viruses. RO viruses. Okay, RO viruses. So the size of viruses, it may ranging from 300 nanometers as TMV to 20 nanometers as RB viruses. Okay. For example, the topmost spectrum, topmost size of virus means largest virus. Okay. The size of largest virus. Very important point. The largest virus size, it is almost equal to the size of smallest bacteria. అర్థమవుతుందా? బ్యాక్టీరియాలో ఏదైతే స్మాలెస్ట్ అని మనం అనుకుంటామో వైరస్ యొక్క లార్జెస్ట్ వైరస్ సైజు బ్యాక్టీరియాలో స్మాలెస్ట్ బ్యాక్టీరియా సైజ్కి సమానంగా ఉంటుంది ఒక వాటిలో. ఇప్పుడు అర్థమవుతుందా? దాన్ని బట్టి ఏమనుకుంటున్నాం, లీస్ట్ ఏదైతే ఉందో బ్యాక్టీరియాలో లీస్ట్, ఆ లీస్ట్ సైజ్కి హైయెస్ట్ అంటే లార్జెస్ట్ వైరస్ సైజు దాదాపు ఈక్వల్ అవుతుంది. సో the size of the largest virus is almost equal to smallest bacterium. Smallest bacterium. Another point, very important. What is smallest bacterium? What is smallest bacterium? That is mycoplasmas. Maybe mycoplasmas. Okay, mycoplasmas. ఇప్పుడు చూడండి, the size of largest virus is almost equal to the smallest bacteria, and smallest virus size is almost equal to the size of ribosome. That is cell of ribosome. Smallest virus size, okay, is almost equal to ribosomal size. Ribosomal size. Very important. About the point. ఏదైతే లార్జెస్ట్ వైరస్ ఉందనుకుంటామో ఆ వైరస్ స్మాలెస్ట్ బ్యాక్టీరియల్ సైజ్కి సమానంగా ఉంటుంది. ఏదైతే స్మాలెస్ట్ వైరస్ ఉందనుకుంటున్నామో such size is almost equal to one of the cell organelle, that is ribosome. That is the size of viruses. Now coming to shape. Okay, shape విషయానికి వస్తే ఏంటి అనేటువంటి విషయానికి వస్తుంది. వెరీ ఇంపార్టెంట్. సో every virus is represented by nucleic acid core. Surrounding this core, that is protein. Such protein is also called capsid. Protein capsid. This protein capsid is to provide not only protection to the nucleic acid, and also it provides a definite shape to the viruses. The definite shape of virus is recognized by, is determined by the capsid. Okay, ఒక సెల్ యొక్క అంటే ఒక ప్లాంట్ సెల్ లేక ఒక బ్యాక్టీరియం యొక్క స్ట్రక్చర్ షేప్ వాల్ ద్వారా డిసైడ్ చేయబడుతుందో, అలాగే capsid it provides a definite shape to the virus, and as well as it provides protection to the nucleic acid present in the virus. There are different kinds of morphological kinds of viruses. For example, spiral helical viruses. There are some helical viruses. What are examples for these helical viruses? These helical viruses, they are long rods. Along maybe they resembles with the long rods. These rods may be flexible or rigid, hard and rigid or flexible. Long rods Some of the best examples for this long rods type of viruses. One is TMV and rabies virus. TMV and rabies virus. For example, this is rabies virus shape. Okay, this is rabies virus shape. Long rods, allowed. Long rods. For example, this is bacteria. This is TMV, long rod, allowed. TMV. Okay. This is rabies virus and TMV. They are a type of helical viruses. They may be flexible or rigid. And long rods. Example TMV and rabies. You know, some of the viruses are polyhedral viruses. Polyhedral viruses. Many-sided. Many-sided. For example, the viruses which attack animals, bacteria, and plants, animals, and bacteria. The viruses which attack plants, animals, and bacteria, they are many-sided. These type of viruses, they are polyhedral shaped viruses. Like this is polyhedral shaped viruses. Okay, like this, polyhedral, many faces, many-sided, polyhedral viruses. Okay, some of the examples for these polyhedral viruses. One is herpes simplest virus. Herpes simplest virus and polio virus. They are examples for these type of viruses. Polio viruses and herpes simple viruses. At the same time, you know, some of the viruses are enveloped viruses. What are enveloped viruses? For example, this is a virus. Sometimes these viruses are also surrounded by protein ఉంది కదా, సెంటర్ లో న్యూక్లిక్ యాసిడ్ ఉంది, దాని చుట్టూ ప్రోటీన్ ఉంది. Surrounding this protein there is a envelope. This is envelope. Such viruses which are surrounded by envelopes, additional protecting layer envelopes, they are considered as enveloped viruses. Some of the best examples for this enveloped viruses. This is influenza virus. These are examples for enveloped viruses. At the same time, you know, some of the viruses, they are also considered as complex viruses. These complex viruses are best examples of bacteriophages. These bacteriophages, they infect the bacterial cells. And these bacteriophages are tadpole shaped viruses. What is the shape of the tadpole? Is represented by a large head and small tail, like this. This is a large head and is represented by a small tail. This is head and this is tail. These type of viruses are represented by two different symmetries. Okay, head is represented by polyhedral symmetry, and tail is represented by spiral symmetry. The combination of this polyhedral symmetry and spiral symmetry, this is considered as binary symmetry. This is called binary symmetry. Some of the best examples for this binary symmetry, bacteriophages. ఇవేం చేస్తాయంటే బ్యాక్టీరియాను అటాక్ చేస్తాయి. బ్యాక్టీరియాను అటాక్ చేసి వాటిపైన ఇవి జీవనం చేస్తూ ఉంటాయి. ట్యాడ్పోల్ షేప్లో ఉంటాయి. హెడ్ ఉంటుంది, టెయిల్ ఉంటుంది. హెడ్లో ఏ రకమైన సిమెట్రీ ఉంది? పాలీహెడ్రల్ సిమెట్రీ ఇన్ హెడ్ అండ్ స్పైరల్ సిమెట్రీ ఇన్ టెయిల్ పోర్షన్. సో దిస్ ఈస్ రిప్రజెంటెడ్ బై ద కాంబినేషన్ ఆఫ్ both polyhedral and spiral symmetries, so that this is called binary symmetry. At the same time, you know, some of the viruses, for example, this is a virus. Okay, some of the viruses, they are also surrounded by glycoprotein capsule, glycoprotein spikes. ఎలా ఉంటుంది? స్పైక్స్ ఉంటాయి. స్పైక్స్ అంటే ఎలాంటి ముళ్ళు లాంటి భాగాలు ఉంటాయి చుట్టూ వైరస్. glycoproteins that forms the spikes in the external portion of the viruses. Glycoprotein spikes. Okay, some of the best examples for this glycoprotein spikes, measles viruses. Emily, measles viruses. కాబట్టి measles viruses లో వైరస్ చుట్టూ ఒక గ్లైకోప్రోటీన్, మామూలుగా ప్రోటీన్ ఉంటుంది, దాని చుట్టూ ఏముంటుంది, ఒక ఎన్వలప్ ఉంటుంది కొన్నింట్లో అన్నాం. ఈ ఎన్వలప్ దేంతో ఏర్పడిన ఎన్వలప్ ఉంటుంది, ఇక్కడ గ్లైకోప్రోటీన్ envelope that forms many spikes in external portions of these viruses. Example, measles viruses. So they are the shapes of viruses. There are different morphological shapes. 1. Helical, 2. Polyhedral, 3. Enveloped viruses, 4. Tadpole shaped complex viruses with binary symmetry, bacteriophages, and also our measles viruses, glycoprotein spikes. Okay, now coming to structure of viruses, very important. ఇక్కడ మనం గమనించాల్సిన విషయాలు కొన్ని ఏంటనేది ఇక్కడ మనకు వెరీ ఇంపార్టెంట్. ఒక రెండు వైరస్ల యొక్క స్ట్రక్చర్ కూడా మనకు డీటెయిల్గా ఇచ్చుకున్నాం. వన్ టిఎంవి, అదర్ వన్ ఇస్ బ్యాక్టీరియోఫేజ్ వైరస్. ఓకే, అసలు ఏంటి ఈ స్ట్రక్చర్లో ఏముంది అనే విషయానికి వస్తే, every virus is represented by a core of nucleic acid. Okay, plus protein. Nucleic acid plus proteins. Okay, this nucleic acid part is called core, and protein part, this is called capsid. This capsid is not a single structure. This is formed by the association of many small subunits. The subunits of these capsids are called capsomers. They are called capsomers. Subunits of capsid. Protein part of the virus is called capsid. Subunits of capsid are called capsomers. These protein parts, it protects the nucleic acid and also it provides a definitive shape to the virus. That is the nature of the protein. Very important. Protein, it not only provides protection to the nucleic acid of virus. Actually, nucleic acid, it is surrounded and packed by protein capsid. It protects the nucleic acid and as well as it also provides a different shape to the viruses. But here, nucleic acid of virus, okay, this nucleic acid may be DNA or RNA, only one type of nucleic acid. There is not the virus with both nucleic acids, only one nucleic acid, either DNA or RNA. Most of the plant viruses that infect the plants, most of the plant viruses, they contain single-stranded RNA as genetic material. And most of the bacteriophages and animal infecting viruses, they contain DNA, double-stranded DNA. So, the nucleic acid may be DNA or RNA. It may be single-stranded or double-stranded. At the same time, this DNA may be circular or linear. ఇలా సర్క్యులర్ గా ఉండొచ్చు న్యూక్లిక్ యాసిడ్, లీనియర్ గా దారం లాగా ఉండొచ్చు, ఎట్లయినా ఉండొచ్చు. ఓకే, సో న్యూక్లిక్ యాసిడ్ కోర్ సరౌండింగ్ దిస్ ప్రోటీన్, దట్ ఈస్ కాల్డ్ capsid with capsules. Only one nucleic acid, either DNA or RNA. Most of the plant viruses contain double single-stranded RNA, and many bacterial viruses and zooposes, animal infecting viruses, they are double-stranded DNA. Okay, double-stranded DNA. At the same time, most of the viruses, they contain only one copy of genetic material. మామూలుగా ఒక కాపీ ఉంటుంది ఒక వైరస్. But some of the viruses, they contain two copies of genetic material. For example, TM, this is one of the best example, HIV. Actually, genetic material of this HIV, this is RNA, but it contains two copies of single-stranded RNA is present in HIV virus. కాబట్టి DNA ఉండొచ్చు, RNA ఉండొచ్చు, అది డబుల్ స్టాండర్డ్ అయినా కావచ్చు, సింగిల్ స్టాండర్డ్ అయినా కావచ్చు, అది సర్క్యులర్గా ఉండొచ్చు లేకపోతే లీనియర్గా ఉండొచ్చు. అది ఒక కాపీ ఉండొచ్చు లేదా రెండు కాపీలు ఉండొచ్చు. మోస్ట్ ఆఫ్ ద వైరసెస్ కంటైన్ సింగిల్ కాపీ ఆఫ్ న్యూక్లిక్ యాసిడ్, బట్ రేర్లీ it is also two copies. One of the example HIV is one of the best example. Okay, now coming to structure of TM. ఇక్కడ చూడండి ఒక TM స్ట్రక్చర్ ఎలా ఉంది అనేది మనకి ఇక్కడ చాలా ఇంపార్టెంట్. దిస్ ఈస్ న్యూక్లిక్ యాసిడ్. ఓకే, దీంట్లో కొన్ని నంబర్స్ వస్తాయి, డిజిట్స్ వస్తాయి. ఆ డిజిట్స్ మనకు ఆబ్జెక్టివ్ లో చాలా ఇంపార్టెంట్, ఎంసెల్ లో ఉపయోగపడతాయి. ఫర్ ఎగ్జాంపుల్ దిస్ ఈస్ వైరస్. ఓకే, this is a TMV virus. TMV virus. This is a genetic material that is single stranded RNA. This is protein and each and every part subunit. This is considered as capsomes. Capsomes. Very very important. But today tobacco mosaic virus was first crystallized by, already know who crystallized tobacco mosaic virus, Stanley, in 1930. Okay, this is a typical plant virus. Plant virus ante daniki automatically untundi single strand RNA. The structure of this TMV was first explained by Franklin. Structure was explained by Franklin. And what is the shape of TMV? Different shapes చెప్తున్నాము. TMV shape, helical symmetry and long rods. ఎలాంటివి? Long rods. ఒకదాన్ని మనం ఇట్లా తీస్తే ఇదంతా పొడవైంది, ఒకదాన్ని మనం ఎలా బ్రేక్ చేస్తే లావు.